Hi everyone, my name is Sarah Gora. I am a first semester master's student in the biology department at UNCG. I work in the Kerner Plant Biodiversity Lab. For my research, I study plant trait responses to nitrogen additions in grasslands, particularly focusing on those below ground traits. As we know, nitrogen has increased globally. The global nitrogen cycle has been greatly disturbed by human activities, resulting in elevated nitrogen deposition in many parts of the world. Human activities produce over half of the global nitrogen pollution. The increasing global food demand is largely responsible for this excess nitrogen, with agriculture responsible for 80% of nitrogen pollution. Industry, shipping, and other factors make up the remaining 20% of global nitrogen deposition. These levels continue to rise, with nitrogen pollution being one of the 5 main impacts of global change. Nitrogen is the nutrient most essential to plant growth, as it is the primary building block of plant proteins. It's an essential ingredient for popular household fertilizers. For example, Scott's fertilizer has a 28% nitrogen content in its top-selling lawn fertilizer. Studies have shown that the availability of soil nitrogen is a critical controller for plant productivity. Nitrogen enrichment on the global scale has been found to cause significant changes in plant productivity, plant community structure, and biodiversity. So this summer, I plan to travel to Kansa Prairie Biological Station in Manhattan, Kansas. Fieldwork for my research will be conducted at 3 ongoing experimental sites at Kansa as part of the LTER network. This long-term ecological research network facilitates comprehensive research focused on the tallgrass prairie ecosystem. This is one of the most productive and biodiverse ecosystems of North America. Specifically, I will explore the effects of nitrogen addition on plant traits in the broader context of its relation to global change effects. This will be accomplished through collection and analysis of plant trait data. Plant traits are defined as any morphological, anatomical, biochemical, physiological, or phenological plant feature measurable at the individual level. So I will be taking above ground and below ground plant traits. These will be collected for my study. First, I will take above ground plant trait measurements like plant height and leaf number. Next, I will then dig up these whole plant samples where these roots will then be washed so I can take root trait measurements like biomass, weight, and branching counts. This is really my main focus. These measurements will then be analyzed to answer my research questions. So the overall aim of my research is to explore how interspecific plant traits vary in response to long-term chronic nitrogen addition. I have 3 supporting questions. First, how do nutrient additions influence root biomass allocation patterns and root characteristics and architecture? To what degree are my 6 sampled species associated with mycorrhizal fungus? And does that association change with nitrogen addition? Lastly, do these below ground trait responses reflect trade-offs between above and below ground traits? Ecosystem services like carbon sequestration, nutrient cycling, and then primary productivity are all in part driven by organismal response, including traits. Below ground traits are particularly significant in this study as they're currently understudied due to the challenging nature of data collection. They're underground. My research will contribute to the broad scale understanding of how plants respond to global change drivers like nitrogen deposition. Thanks for watching.